For those of you who are new to us, Cure Parkinson's is a medical research charity. We were founded in 2005, and we are very, very focused in our remit. All we really focus on is funding and facilitating and supporting research focused on disease modification and Parkinson's, with urgency for people living with the condition. And we do our research funding primarily through 4 calls every year for applications, grant applications for preclinical and clinical research. And the primary category, well, the primary criteria for these applications, they have to be of disease modifying nature, and preclinically they must be within 5 years of clinical testing. So we're looking for that proof of concept before we go to a clinical trial in people with Parkinson's. And we also, because we have this wonderful independent committee that evaluate all of our grants for us, and we have no influence over that committee. We also like to invite applicants to submit their applications for review by the research team at Cure Parkinson's. We can provide feedback before they formally submit, and that way we're not wasting the committee's time and we're not wasting our applicants' time. On the clinical side, we also have the International Linked Clinical Trials Initiative, and this is a large committee of 25 world-renowned experts that we gather every year for a 2-day face-to-face meeting, at which we present them with a set of dossiers. Each dossier is everything we know about a particular drug. There must be some evidence for potential disease modification with these drugs, and we ask the committee to review those dossiers and rank and prioritize which ones should be going forward for clinical testing. And that program's been going for 13 years now, and it's resulted in a large number of clinical trials. Where you see the CP logo is where CP has been a direct funder, with our funding partners, the Van Andel Institute in Grand Rapids, Michigan, who help coordinate the program with us. And I'll draw your attention to one trial in particular here, which is the nicotinamide riboside NOPARK phase 3 study, because Harris won't be talking about that today, but he is the coordinator of that trial, and that will be reading out early next year. So that'll be something to look out for in the new year. So the objective for us as a charity is to slow, stop, or reverse Parkinson's, to alter that trajectory of the progression of the disease. And we're looking for disease modifying therapies or interventions for Parkinson's. And we have outlined this in our new updated research strategy. Next slide, please. That's the cover of it. You can find them in physical form outside. You can also find them on our website as well. Today is the launch of the new research strategy, new updated research strategy. And the background to this is that as a member of the AMRC, which is the Association of Medical Research Charities here in the UK, the association requires us to every 5 years, to have a review of our research strategy and make an update, any adjustments that are required. And this is where you pull in members of the community that you're dealing with, both the researchers, the stakeholders, people affected by the condition. And you present them with your current research strategy, you present them with some ideas about where you'd like to take that research strategy, and then you have a discussion and Q&A and really try and nut out what's the best approach. And taking some of the feedback and some of the discussions from that workshop that we held last year, we've used that to help shape this new research strategy. Next slide, please. And there are 4 basic pillars to the research strategy. The very first is to grow our treatment selection program. That is the ILCT program we were talking about before. So we're trying to expand on how we identify these agents that could potentially be disease modifying. And then the second pillar is to accelerate the clinical testing of those drugs or interventions. The third is to really double down and explore how we can increase our chances of identifying properly disease-modifying therapies in the form of combination therapies. Everything we've tested up to date has been a monotherapy approach, just testing a single drug in a single trial. How can we improve on that? We could test two drugs side by side, and then in combination, if there's a solid rationale for why they could possibly work together. And then the fourth is the making disease modification more personal. So personalizing and targeting the right people with the right drug. So expand the identification of new therapies, accelerate the clinical testing of those therapies, double down and really increase our chances of finding a new therapy with combination therapies, and then try and personalize it to each individual person, target the right people with the right drug. Next slide, please. So I'm going to take you through each of these pillars firstly and just talk about some of the tactics that will be applied to each of them. So in the growing of the treatment selection program, you might be thinking, how do we expand on what we're already doing? Next slide, please. One way we're looking at doing this is exploring cross-disease collaborations. So this is where there might be two diseases where there's overlapping biology, and we'll work with the people that work in that field and help working together, pooling the expertise, pooling the resources. We'll explore how we can identify drugs that modulate the biology for both diseases, and whether we can start clinically testing some of those drugs. And we're already doing this with Alzheimer's Research UK. So in Alzheimer's and dementia, there are biological processes that overlap with Parkinson's. For example, there are proteins that aggregate together. And Alzheimer's, one of those proteins is a protein called tau. And in Parkinson's, there's also aggregation of tau in people with affected by the condition. So rather than working in isolation, why not work together with Alzheimer's Research UK and pull those resources together, pull the expertise together, and explore drugs that are affecting, that could potentially be modulating the biology, and test those in partnership with them. Next slide, please. And also have more international collaborations. At the moment, we're very UK-centric, and we're looking to really make ourselves geographically agnostic and take advantage of situations in particular countries where there are specific types of Parkinson's, or there's specific resources available that we could use in this effort. Then also we're looking at de-risking early stage agents. Some of these dossiers that we write are for molecules that aren't quite ready for clinical testing. And you'll be learning a lot more about this from Harris when he talks about Slepnia. This is a platform that's going to be accelerating the testing of these early stage molecules in cohorts of people with Parkinson's. So very early phase 2B, phase 2A, looking at safety and tolerability, target engagement. Is the drug actually hitting the target you want it to hit? And is it getting into the brain? These sorts of tests. Next slide. Oh, one of the detail there was the ILCT accelerator program, which we have as a special tool for the ILCT committee members, where they think a molecule is not quite ready for clinical testing. They can commission us to, they can tell us to go out and commission a piece of research. They might think this is an interesting drug, very interesting biology. We'd like to see it in people with Parkinson's, but before we do, we need to see this, this, and this. And using that program, we go out and commission the research to get it done as quickly as we can. So there are different ways that we are expanding the identification of new molecules of interest. And then we're accelerating the clinical testing of new therapies as well. And one of the ways we're doing that, next slide, please, is through the supporting of different clinical trial designs. And at the moment, the main one we're focused on multi-arm, multi-stage clinical trials. These are large platforms where you're testing multiple treatments, treatment 1, treatment 2, and treatment 3 against a single placebo arm. And the beauty of this is that that's the multi-arm part, but the multi-stage is that you are having interim analyses throughout the testing, continuously analyzing the results. Rather than waiting till the end of the study, you're looking at the results as the study is going on. So you can determine that treatment 2 and treatment 3 in this particular example aren't working. So why continue with them? Take them out and bring in treatment 4 and treatment 5. Treatment 1 is showing signs of doing something, so we'll keep testing it. And you can use this dynamic approach to accelerate the clinical testing. You can go from phase 2 all the way through to phase 3. And once a molecule is found to be working in people with Parkinson's, could potentially go through the regulatory process and get approved, and then that becomes standard of care. So everybody in the rest of the arms will go on to that treatment as standard of care. And so it becomes compounding, if you understand what I mean. So that's going to be discussed this afternoon, both Paris and Georgia, and Shireen will be talking about that. Next slide, please. This sort of gives Sleipnir away a little bit, but it is another method of accelerating the clinical testing of molecules of interest. Sleipnir is going to be a 3-month trial platform, so 3 months of testing particular drugs, and the focus will be on safety and tolerability over that period of time in people with Parkinson's. Is it getting into the brain, and is it hitting the target? Is it doing what we want it to do as far as biology goes? And one note here for people observing online, or people in the audience who are involved with research, is that we definitely need more of these Sleipnir like platforms, because going forward there's going to be a great thirst for molecules for testing on NAMS platforms. Next slide, please. And speaking of those other platforms, so we've got the EJS platform, which is getting rolled out here in the UK, accelerating clinical trials in Parkinson's. Harris might talk about the Hydra platform. This is similar to EJS phase 3 platform. We've got platforms being set up in France. There's a multi-arm platform already underway in Australia. And then you have the Sleipnir project, as I said before. The most ambitious of these efforts is coming from the States, which is the P2P trial. And this is being run by the Michael J. Fox Foundation. And what they're trying to do there is they're trying to test interventions before people are diagnosed with Parkinson's. So this is called the prodromal phase of Parkinson's, where you're showing signs of being at risk of developing the condition. And the team at Fox are taking on this ambitious task of running clinical trials in people that aren't necessarily sick yet, or they're not showing the signs of Parkinson's yet, but there's hints that they're going in that direction, and they want to start testing molecules and see if they can stop those people from ever actually being diagnosed with Parkinson's. Next slide, please. Third is the championing of the development of combination therapies for Parkinson's. Next slide. Thank you. So combination therapy is not a novel idea for Parkinson's. Since the 1970s, the gold standard treatment for Parkinson's is Senimit, which is a combination therapy. This is a symptomatic treatment, not a disease-modifying therapy. But the concept is very easy to understand. It's made up of 2 drugs, firstly levodopa, but also another one called carbidopa. Levodopa is the key ingredient in the production of dopamine, and it uses an enzyme called dopamine decarboxylase to be converted into dopamine. Now, carbidopa blocks dopamine decarboxylase everywhere in the body except the brain. So as a result, if you just gave somebody L-dopa by itself, levodopa by itself, it would get converted into dopamine in the gut primarily, and they'd probably feel a bit sick, they'd feel a bit nauseous. In combination with carbidopa, you're blocking that conversion. So the only place in the body that the dopamine can be converted is in the brain. And as a result, we have a treatment for Parkinson's. The dopamine is being made where it's required in the brain. And it's this kind of synergistic effect that we're looking for in terms of potential combinatorial approaches for disease-modifying therapies in Parkinson's. It could be that one drug has shown signs of doing something interesting in the past, or it shows it's neuroprotective in models of Parkinson's. But what we're looking for is what else could you put with that to really hammer home the effect. If you're going to win at something, why win by a little bit? Why not win by a lot? And that's where we've asked, next slide, please. Well, before I jump to that, the basic concept behind the clinical testing of these drugs would be pretty straightforward as well. At the moment, we test placebo and treatment A, just the two arms of a study. In a combination therapy approach, you'll still be testing the placebo and you'll still be testing treatment A by itself, but then you'll be throwing treatment B in as well. And there'll be a rational reason why you would put the combination together as well in a fourth arm. So instead of having two arms to your clinical trial, there'll be four arms, larger clinical trials, but hopefully that combination will demonstrate itself to have more of an effect than either of the drugs by themselves. Next slide, please. So we've asked the Cure Parkinson's trustees whether we could have an open funding call for rationally designed combination approaches for disease modification. And we're looking at preclinical and clinical projects. And they've agreed, and they've ring-fenced £2 million for this effort. And the funding call opened on the 1st of October. Rachel Hughes, who's here today, has been driving this. There she is in the front row, on the research team. And we're really looking forward to, we're really curious as to what sort of approaches we're going to get. We've had a lot of enthusiasm thus far from the community, the research community. We've had a couple of Q&A sessions explaining the program and addressing any questions that they might have. And thus far, it's been a good level of feedback. Next slide, please. In addition, we've put together a special committee of experts. People have got experience in the testing of combination therapies, but also a bit of knowledge about Parkinson's. And key among them is Professor Fiona Dockered, who is the chief scientific officer at Alzheimer's Research UK's UCL Drug Discovery Institute. And she'll be chairing this panel. And we were quite touched by one of the comments that came in from one of the panel members who applauded us for charting this area of research. Next slide, please. And finally, we're going to try and make disease modification more personalized. And here we're still exploring exactly how we're going to do this. Next slide, please. But a great deal has already been done by others and ourselves in terms of the genetics and the biomarker and wearable space. And what we're really looking to do here is not so much stratify different types of Parkinson's, but stratify in our clinical trials who will respond to a particular drug and who won't. So here we're thinking more along the lines of clinical trial in a dish before we start the clinical trial, and see if we can't pull groups that would respond to a particular drug and test them either with a placebo or that particular drug. So here I would just say, watch this space. Next slide, please. So just to summarize, the goal here with our updated strategy is to grow the treatment selection program, expand it, take as many ideas as we possibly can, then accelerate the clinical testing of those new ideas, champion the development of combination therapies. Why test one when we can test multiple drugs at the same time? And finally, personalize it, make sure we're targeting the right treatments to the right people, and that should be the testing ground for these new therapies. And with that, I'll say thank you very much, and I'll take any questions. Next slide, please. Thank you.
Sorry.
Oh, question here.
I'm intrigued.
There's a microphone, sorry.
Thank you. I'm intrigued how you allocate capital between those four themes.
At the moment, we've got a lot of capital invested in the ILCT program, and the projects that all of the projects that are funded go through the AMRC approved process of having a two-stage review process and peer review feedback in the middle. And it is a question for the trustees as to which projects they actually fund. They will take the recommendations of our grant evaluation committee and use that to make their funding decisions. In addition, we also are now having the open funding call with the panel for the combination approach. So it's a separate committee, but all of this is in line with the AMRC funding guidelines. Does that answer your question, or?
Yes, thank you.
Forgive me because I came in a bit late. I might have missed it. But not thinking about therapies, but thinking about cure. Have you done any work in that direction, in finding causes and finding the cure? Thank you.
So usually with these research update meetings, we will provide an overview of the clinical trials that are ongoing at the moment. Given that today is the launch of the updated research strategy, we're focused more on that. You're going to hear about some clinical trials that are ongoing at the moment with some of the other presentations. As I said, Harris might want to talk about the nicotinamide riboside study. It's a phase 3 study. So that's the last stage in the clinical testing. And the results will be out early next year. And in addition to that, there are a lot more studies. I had a slide outlining the clinical trials ongoing for the ILCT program, and there's a lot there that's going to be reporting out in the next 18 months.
Sorry, this is quite a general question, really. Why do you think it has taken so long? And I'm not blaming CPT in any way for this. In many ways, they're in the vanguard of the changes. But why do you think it has taken so long to recognize that clinical trials have to reflect the majority of the population, which is not drug naive? They're essentially taking combinations of drugs already. So to look at that seems obvious to me that it's a very good way forward. But why has it taken so long?
I think, to be fair to the research community, safety is always first and foremost consideration. So there is a risk with going down the combination therapy approach. Everything works wonderfully preclinically, but once you start looking at it in humans, there could be side effects popping up that were unexpected. But yes, a lot of people are already self-experimenting with different combinations of things. But we need to be doing that in a structured fashion going forward in clinical testing. Rory, that is hand up.
I'm fantasizing here. If you suddenly heard that you'd come into a huge sum of money, besides going off to Bermuda or wherever, what would Cure Parkinson's spend it on? We always hear about, for instance, HIV and what a brilliant result there was with that, and that was a wall of money. What would a wall of money do to Cure Parkinson's?
If anyone else has easy questions, please feel free to take the microphone. Now, if we had, so this is the $1 billion question. If Elon Musk or somebody walked in and said, "Here's $1 billion on the table, what are you going to do with it?" It would be a combinatorial approach. It would be very, very personalized, because we're dealing with a heterogeneous population of people affected by the condition. And you really need to get down to not N of one, but N of very few. And look at, focus on treatments that will help those small groups of people. Parkinson's has basically been this big basket that we could just put people into because they share resting tremor, slowness of movement, and rigidity. But increasingly, we're seeing that it's not as simple as that, and we should be orienting our clinical testing towards those specific subpopulations. So it would be very much focused around that, and it would be, as far as I'm concerned, combinatorial. I'm going to ask Tom if he's got any thoughts on this matter, because I can see he's desperate to get involved. And I might bring Harris in as well, just to make it interesting. Somebody feel free to throw a chair if they don't like what they have.
Okay, Rory. So one of the most important things to measure in Parkinson's is the protein alpha-synuclein, which we think is not the only cause, but it's a major contributory cause to Parkinson's disease. And if we could do a scan that could image alpha-synuclein, that's a really good way of diagnosing it and monitoring how it spreads over time and whether a treatment actually makes a difference to its progression. And there are investigations which are becoming advanced, which are optimistic, looking at an alpha-synuclein quantitative PET scan. So the Fox Foundation have been trying to look for this for years, and there's significant progress being made. So more money into that would be good. The other way of diagnosing Parkinson's and measuring alpha-synuclein is to take some spinal fluid and look for alpha-synuclein in its abnormal forms. So if we could do that with a blood test, that would be really important, and quantitate it to see is not just whether it's present or absent, but how large the dose or load of that problem is. And then, of course, Parkinson's is complicated. So it's not just alpha-synuclein, it's lots of other things. And the lady's alluding to, other environmental factors which can influence the risk and rate of progression of Parkinson's. And because there are so many complicated contributory factors, thinking about environmental exposures, diet, behavior, toxins, pesticides, then we need to start thinking about big data and AI approaches to investigate how people behave, what they're exposed to, what their risk is for them. And then you can throw in a little bit of extra data from genetics, a little bit about maybe their gut microbiome, a little bit about what their phone tells you about where they're going to and how often they're doing certain behaviors. And then you can get a really good understanding. And someone like Elon Musk, if his heart was in the right place, that kind of funding and that kind of expertise would be really helpful.
We're not sure if his head, we're not sure if his head's in the right place at the moment.
Yeah.
But I'll invite Harris to come up with stage. Now, we're way behind schedule, but this is an interesting discussion. And I'll ask him to address this question. Maybe if you were offered $1 billion, what would your approach be? Because he is coming less from the alpha-synuclein approach and more from a mitochondrial approach.
Right. So the $1 million question. So I think, first of all,
billion.
Yeah, that's what I said. $1 billion. Yeah, $1 billion. Elon Musk, more than $1 billion, several billions of dollars, right? So just for the purpose of the exercise, I'm going to take a different road. So I fully agree. I fully support what Tom said, and that is the holistic approach. And I think that's what we need to do. But, so I don't repeat what Tom said, I'm going to take a different approach. I'm going to take the reductionistic approach and say, I'll exchange Elon Musk's money with a genie. The genie in a bottle, right? So I rub the lamp, whatever it is, and the genie comes out and it gives me one wish. But the condition is it cannot be the goal. So I can't just ask for the cure. It's not that easy. It can be means to the goal, right? So there's one thing you can have, Harris, the genie says. What is it going to be? So now I have to choose. I can't say everything like Tom said. I have to choose. For me, today, I would have chosen an objective, accurate measure of disease activity. A way for me to objectively measure the disease so I can do trials more efficiently, fewer patients, faster, and actually be able to trust the results that I see. Get rid of the tremendous placebo effects that we have in Parkinson's and the likes of that. So that would have been my, I think, wish to the genie. And it's not an easy answer, right? So if you had asked me the same question a couple of years ago, I would have probably told you a stratification marker to be able to break down Parkinson's into subtypes. So, it's not easy. But I think right now, I would have gone for the marker.
Good answer. Very good answer.